Meet Benji, a 2-year-old collie. Benji has been displaying symptoms of loss of balance, tremors, and losing weight. He has a healthy lifestyle, and his owners make sure he gets proper nutrition and exercise. He has Frontline every 8 weeks and Heartgard every month. Both of these medications were recommended by a vet. 6 months later, his same symptoms got worse and he started having seizures about twice a week. After days of vet appointments and wondering what was wrong with Benji, the vet finally diagnosed him with an MDR1 gene mutation. The vet took a sample of Benji's blood and sent it into a lab to get tested. First, the DNA is separated from the cells and many copies are made. This happens because of a method called polymerase chain reaction, aka PCR. PCR uses the enzyme Taq polymerase to make specific parts of the DNA larger. The DNA molecules are then split at specific locations and the code is further looked into. After looking at Benji's code, it was shown that he had this mutation.
Meet Marley, an Australian Shepherd and Border Collie cross. She has a 50% chance of getting the MDR1 gene mutation. Luckily, Marley's vet was aware of this possibility and diagnosed her as a puppy. The preventative treatment for Marley is to use the drug selmectin instead of Heartgard. Studies have shown that dogs with the MDR1 gene mutation do not react to the drugs with selmectin in it. Marley can live the rest of her dog days with no complications because she was diagnosed early on.
Meet Bear, a German Shepherd. After hearing about Benji's troubles and learning that German Shepherds have a 10% chance of having the MDR1 gene mutation, Bear was tested. He was lucky enough to be born without it and he won't need any treatment. Now he can live a long and happy life doing what he loves, hunting criminals.
Bad boys, what you gonna do? What you gonna do when they come for you?
According to Washington State University, the MDR1 gene encodes P-glycoprotein, a drug transport pump that plays an important role in limiting drug absorption and distribution, particularly to the brain, as well as enhancing the excretion of many drugs used in dogs. Some dogs, particularly herding breeds, have a mutation in the MDR1 gene, leaving them defective in their ability to limit drug absorption and distribution. These dogs also have delayed excretion of drugs that are normally transported by P-glycoprotein, making them susceptible to severe drug toxicity.
The MDR1 protein transporter, also known as the P-glycoprotein, is made up of a phosphoprotein that contains 1,280 amino acids. PGP is made up of 2 ATPase, which is split equally. At each ATPase site, glutamate residue replaces a catalytic base for ATPase hydrolysis.
The mutation of catalytic glutamate affects the ATPase activity and slows it down, causing the specific drug to not flow through the PGP as fast, which causes buildup of the specific drug.